Hello everyone, I'm going to be sharing with you guys my review paper on CAR-NK therapy, which I've written to show the specific biology behind NK cells and also the process behind how you design a therapy like CAR-NK therapy. So let's move on right into the article. So as you can see here on the screen, this is my article which I wrote on Medium, and you see the title of it is The Fundamentals and Innovation Behind CAR-NK Therapy. I'll leave this title in the description if you want further reference. And here. So we'll move on to the first part, which is my abstract. So this is really like a synopsis of what I've written. And if you want to like see what I've written in a short amount of time, then this is what you want to read to see if you're actually like interested in reading it, or if you don't have enough time to read the whole article. It is pretty lengthy. Yeah, this is where you would want to go. So let's move on to the table of contents. So this is where you'll see all the different sections, and then introduction. This basically gives a brief introduction to what I'm writing about. Not like the abstract where it includes all the different things in my article, like a summary, but it's more of a like it introduces the topic so that you'll understand when I go into the first part. So speaking of the first part, that is NK cell receptors. So I speak about the different types of NK cell receptors because these cells have different ways of seeing like which cells are our cells and which cells are other cells that we don't want. Okay, so the first part, 1.1, is KIRs, which is killer immunoglobulin-like receptors. And then see, I have a little picture here. This is what the actual shape of the receptor is. And this is basically a lot of biology and signaling pathways behind it. And then basically, we show the difference between an activating receptor, which is what I spoke about above, with an inhibitory receptor, which is what I spoke about here. And after that, we'll move on to a different type. Still pretty similar, but it's still a different class of receptors called CLRs, or C-type lectin receptors. So this is basically what I've shown, and this is a famous CLR, which is NKG2D. It has a very specific role to like say sense different ligands like MICA and MICB. So, and then we move on here. So this is basically a diagram, and I'll include these diagrams that I've gotten from the research papers, which I'll show in a little bit. So I've shown this diagram to to show the pathway between NKG2D and how it actually leads to these different things. And these different things are brought up here in the next section, part 2, which is actually showing the NK cell cytotoxicity and how it kills the actual cells that it's already detected from the first part. So here we see the different parts, like what are lytic granules. This is more of like an introduction to how the NK cell actually like kills, what's in those little vesicles that it compacts and sends out. And then we go on to the formation of the immunological synapse. This is an important process as it's actually where those little lytic granules will be released. And there's different parts to that, like localized actin polymerization, adhesion to the target cell, and then you also have the second part is the movement of the lytic granules. So this is a process that's happening simultaneously with the formation of the immunological synapse, and this is basically where you get your MTOCs, or microtubule organizing center. You might be familiarized with this already with the centrosome, which is used famously in mitosis if you're in grade 10. Um, and then you get to the next part, which is docking and fusion with the plasma membrane. This is actually where it's released. And then finally, we have this huge complex diagram, which basically shows a lot of what I've covered here. There's some stuff that I haven't covered, but basically it covers all this, and it's really nice. It's a nice visual to really see what's going on. And we get to part 3, which is failure of NK cell surveillance. So this is where you'll start to see cancers form, and this is the process of that. So I go into a lot of details about the tumor microenvironment here, and basically there's 3 specific topics. There's presence of immunosuppressive molecules. So one of these is TGF beta, basically inhibits NK cell metabolism, and next, we got change in ligand expression. So these are basically reducing the amount of ligands that are present on NK cells and cancer cells themselves. So cancer cells will show less inhibitory ligands, and NK cells will, after being signaled, will show less activating receptors. So then you go into the next part, which is this is a famous part of the tumor microenvironment, overregulation. So this is where you start to see 3 different types of cells. Tregs, T regulator cells, and then myeloid suppressor derived suppressor cells, or MDSCs for short, and then finally tumor associated macrophages. And then this is where you actually get, this is the part of my article where I go into CAR NK therapy. And so you see here, this is a little introduction to it. And then here is the actual function of CARs. So I give three different expansions into why a CAR works while other receptors don't. And then we go into the parts of the CAR, which is also important because, like, when you design this CAR, it has to fit certain requirements to actually work in an NK cell. You can't bring over different elements from like a red blood cell and then a liver cell and then just add them into a CAR. You need to actually understand the signaling pathway, which is what I went over in the first part. So this part is not it's not completely artificial, as in like we're just designing whole things. We're actually just taking different parts from other receptors, but increasing their vivacity and increasing their potency inside the cell. So you have the extracellular domain, the transmembrane domain, and the intracellular domain. And here I also go into the evolution of CARs and how they've changed over the years. Even though it's a fairly new treatment, it's actually very complex and has gone through several changes. Here are the four generations that are presented in this research paper I found. And then finally, we have 4.2 plasmid design for part 4. And then this, it goes into viral vectors and how these CAR treatments are actually administered. So there's a lot of writing here, and this is an EGFP, which is a fluorescent protein, which scientists can use to actually see how these CARs are being expressed on the different NK cells. So you want to see, like, oh, is this protein being expressed? Because you don't want to just inject normal NK cells into a person. You want to actually inject the CAR because that's what's going to be able to treat the cancer, right? So the EGFP is used to see if that CAR has been expressed on the NK cell. And then this is the last part, part 5, which is sources of NK cells and clinical trials. So this is more of a conclusion part, which basically goes into how these NK cells are sourced, because you can't just like spawn them out of nowhere. You need to get them from somewhere, right? So here are the different parts, and here's a little diagram showing all the different pathways of where you can get these NK cells from. An example of this is iPSCs, which are pretty cool. The researcher that found these actually won the Nobel Prize, um, and basically taking normal cells, turning them into stem cells, and then turning those stem cells into NK cells. That's a process that we use. There's still disadvantages of each of these processes, but we're working to fix these. And finally, we have clinical trials. So these are three clinical trials that I show that are actually in the progress of going right now, which basically this is not, it would not be possible to have these treatments without actual valid proof behind them, and this is what these clinical trials are trying to show. And then discussion, this is where I go into more, this is my conclusion to the review article. And finally, we have the sources. So it would not, obviously, this research paper would not be possible without all these research papers that I've shown here. And I encourage you to actually take a read of all of these, as they're really good. And sometimes I can't fit all the information from these papers into my own article. But yeah, so let's go back, and thank you for watching, and I hope to see you guys next time. Thank you. Bye.